All right, ladies and gents, welcome back. So today let's take a look at actually putting to the test some of the information we've been talking about with the idea of osmosis. Sometimes I find that kids actually make better connections when we actually start putting some numbers to things. So let's try some practice problems today. So for each of the problem sets that we're going to work on, there's a couple of things that we're going to remember. So we're going to have a sort of pseudo cell, a permeable membrane that's placed into a beaker that's going to contain certain dissolved substances inside that membrane, as well as things outside in the beaker itself. So our membrane is going to be permeable to things like glucose, potassium, sodium, water, and iodine. All right, that's what we're looking at right here. Now remember that that term permeable means that things can pass through. So glucose can go through the membrane, potassium can go through the membrane, as opposed to being impermeable. In this case, it's going to be impermeable to that starch. So that means starch can't go through the membrane. Something else that's going to come up in a particular question is just this idea that when you mix iodine and starch together, you actually get this bluish black color. So iodine by itself is sort of yellow, the starch is white. And when they mix together, they go through a chemical reaction and create this bluish black color change. Something else to keep in mind as we're working through these problems is the idea of what does isotonic, hypertonic, and hypotonic actually mean. So if we review what we were talking about in the last set of notes, that prefix iso means that things are going to stay the same. So when I look at water movement, there's no net movement, meaning that the water is moving in and moving out at the exact same rate, and so they're basically canceling each other out. If I have 10 molecules of water moving into my cell, then I also have to have 10 molecules of water moving out, and so if I balance out that what's on either side of the cell, it basically nets or results in zero. There's nothing being added, there's nothing being taken away. And so as a result of that, your cell shape is going to stay the same. In a hypertonic solution or environment, remember that hyper means more, there's more solute outside the cells than compared to inside. So as a result, that water gets sucked out. It's being pulled out, it's being moved out of the cell. And so your cell is shrinking, it's being dehydrated. Okay. Hypotonic, on the other hand, means that there's less solute. So less solute outside my cells means more water. So what ends up happening, that water then moves into my cells, the cell swells up. All right, so let's try this. So if I take a look at problem set A, the first thing we're looking at is this scenario. I've got 90% glucose outside of my beaker, 10% glucose inside. So if all I want to take into account right now is that glucose, first question I want to ask myself, is this particular membrane permeable to glucose? So go back to what this says. My membrane is permeable. There's glucose. It is permeable. So yes. All right, that membrane is permeable to glucose. Now that I know that that glucose can go through the membrane, the next question becomes, will that glucose diffuse? Well, are the percentages of glucose on either side of the membrane identical? No, I've got 90% outside, 10% inside. So that means that they're going to end up diffusing till they can balance each other out, until they can get to a state of equilibrium. So this is an example of passive transport. All right, that glucose is going to move from where it's high to where it's low. So right now, where it's the highest, it's that 90% in the beaker. It's lower inside that pseudo cell. And so, yes, that glucose is going to diffuse. Where is it going to go? From an area of high to low concentration. So from that 90% glucose to that 10% inside the cell. So it's going to move in. Now let's take it from a standpoint of osmosis. All right. Well, these are solutions, right? So any solution has to add up to 100%. So if I take a look at the amount of water inside my cells, well, currently of that 100% solution, 10% of it is being accounted for by glucose. So what does that mean? Well, 100% solution minus the 10% that's being accounted for the glucose means the rest of the solution must be water. And so I've got 90% water inside my cells. Let's go out into the beaker. Again, 100% solution, currently 90% of it is being accounted for by glucose. So 100 minus that 90% from glucose leaves us 10% water outside my cells. So now if we assume that glucose isn't going to move, we're just worried about the water, Is osmosis going to occur? Well, take a look at the percentages of water. Are they identical inside my cell versus the beaker? No, I've got 90% inside, 10% outside. All right, so yes, osmosis is going to happen. Then the question becomes, is my cell going to swell up or shrink based on the way that water is going to move? Well, remember when we talk about osmosis, this is still passive transport. So that water is going to move from where the water is high to where the water is low. Well, right now, my water level is much higher inside my cells. I've got 90% water inside that cell. So that water is going to move from where it's currently high to where it's currently low. So what does that result in? The water rushing out of the cells, and if I deflate the cell, it's going to shrink. All right. So final question then becomes, is the solution in the beaker hypotonic, hypertonic, or isotonic? Again, we're assuming that those solutes aren't going anywhere. So right now I've got a 90% glucose outside my cells, 10% water. Water is being drawn out of the cell because of that high glucose number. So remember, a higher number, hyper means high, and so my cells are currently in a hypertonic environment given this particular set of conditions. If I take a look at problem B, now let's make it a little more complicated. So I've got 60% glucose, 10% starch in the beaker, 10% glucose, 60% starch in the cell. But let's take it one step at a time again. So my first question, is the membrane permeable to glucose? Can glucose pass across this membrane according to the set of parameters we've been told? And the answer is yes. So is the glucose going to diffuse, and then in what direction? Well, this is still passive transport. So it's going to go from where it's high to where it's low. Where is it currently higher? That's 60% outside in the beaker, so it's going to move to that lower percentage inside the cell. Now I've got starch on top of it. So if I go back to that initial set of instructions, did it tell me that starch is permeable? And in this case, it's not. So is the starch going to diffuse? Well, not really, because it can't go through the membrane. So it's sort of a not applicable question. It can't diffuse. So will it? No. All right, and so it really doesn't matter which direction, even though the numbers aren't identical, even though there's 60% starch inside, 10% outside, it really doesn't matter because the starch is just a big carbohydrate. It's too big, it can't pass across that membrane, and so it's stuck where it is. All right, let's take it from the osmosis standpoint again. So how much water is inside my cell right now? Well, 10% of it's being accounted for by glucose, 60% is being accounted for by starch. Remember, I have to add this all up to 100%, so what's left over to be water? 30%. Go on the outside, same idea. 60% glucose, 10% starch, so what's left over to make that 100 to be water? It's another 30%. So now the question is, does the osmosis occur? Assuming my solutes don't go anywhere, the glucose and the starch stay as they are, all I'm dealing with is the water. Well, it kind of depends on how you want to answer this question. Will osmosis occur? You could tell me no, right? Because the number percentage of water is 30% inside, 30% outside, so they're identical. You could also tell me yes, but then you would need to clarify the fact that the water is going to move in the same direction. So will the cell stay the same? Yes, because the water is going to move in and out in the same direction. So the amount of water moving into my cells is equal to the amount of water moving out, so there is no net movement of water. So if we're going to stay the same, that means we're dealing with an isotonic solution. All right, so let's try this one. So I've got potassium, sodium, and starch inside my cells at 10% apiece, 100% water outside my cells. So let's take it again, one solute at a time. So is the membrane permeable to potassium? We're told it is. So if I've got 10% inside my cells, there's nothing written outside except for 100% water. So what does that mean? There isn't any potassium at all. So 10% inside my cell, 0% outside my cell. So what does that mean? It's going to move out. If I go to the sodium, is it permeable? Yes, it is. And again, 10% inside my cells. There's nothing written outside, so it's understood to be a 0. So once again, the sodium's going to move out as well. Is the membrane permeable to starch? No. So is it going to diffuse? Doesn't really matter what the percentages are because the starch can't go across the membrane. All right, let's take it from the standpoint of that water again. Now I've got 10% potassium, 10% sodium, 10% starch inside my cell, leaving me with 70% left over to be accounted for by the water. I've got 100% water outside my cells, so that's already done for me. All right, there is no solute at all. It is just pure water. So, do the numbers equal out? Is there the same number of water inside and outside? No. So will osmosis occur? Yes. Now that water's still going to move from where it's high to where it's low. So what's that going to result in? Well, I've got 100% inside, 70% inside. So that water's going to rush in. My cell's going to swell up. All right. So what does that put me into? This is a hypotonic solution. All right, let's take a look at problem set D. So I'm told 38% glucose, 15% sodium, 3% potassium, 5% starch. That's all in my cells. Outside my cells, I've got 40% glucose, 20% sodium, 1% potassium. So first question just is, which solutes are capable of crossing? Which of these materials are able to go across my membrane? All right. So when we go back to that initial set of instructions, what does it tell me? Well, it tells me that the glucose can pass, the sodium can pass, the potassium can pass, but the starch cannot. All right, so starch isn't going to move, the rest of these are, so which direction are they going to go? Well, if I take a look at the glucose, I've got 40% outside, 38% inside. It's going to go from where it's high to where it's low, even if it's not a huge number, so it's going to move in. Your potassium's also going to move, your sodium, your Na+, is also going to move in from 20 to 15%. Your potassium, on the other hand, is going to go the other direction. It's higher inside the cells than outside, so it will move out. All right, again, let's take a look at the water percentages. So if I add up everything that's in my cell, subtract it from 100, that leaves me with 39% left to be part of, or to be accounted for by the water. Outside my cells, I also have 39%. So will osmosis occur? Again, this question depends on how you answer it. You could tell me no, so long as you understand that those water percentages are identical, and so the net result will be no water movement. That being said, although your cell will stay the same shape, the water movement going in and going out is still happening. It's just happening at the same rate, and so it's balancing itself out. So what am I dealing with in this case in terms of a solution or an environment that cell is in? It's another isotonic situation. So let's take a look at this idea. So same setup as problem D with the solutes in and out of the cell, but now let's take a look at a pretty classic example of identifying how molecules are moving. I can't see any of this by just staring at a beaker and a cell or a membrane in a beaker. So what happens? Well, a common way to test this is to use starch and iodine. All right, so starch is white. Iodine is yellow. I mix them together and they're going to turn this sort of purplish black. They go through a chemical reaction and they cause that color change. So the first question here is, if I add iodine to the beaker and I wait a few hours, I come back to that beaker and I check what the color of the solution in the beaker is, what should it be? And your answer is that the solution in the beaker should stay yellow. There's no starch in that beaker. I've got glucose, I've got sodium, I've got potassium. The starch is all in the cell, but remember that the starch is impermeable to that membrane, meaning it can't pass it, meaning it can't get out of the cell. So I can put as much iodine into that beaker as I want. It's never going to come in contact with the starch in the beaker itself. And so it's going to stay that sort of yellowish color. Now let's take it from this side. So I add the iodine to the beaker. I wait a few hours again, but now rather than checking the color of the solution in the beaker, I'm checking the color in the cell. This time, is it going to stay yellow, or am I going to get that purplish black reaction to occur? And in this case, I will get that purplish black inside the cells. The iodine is permeable to the cell, so that iodine will move from where it's high outside in that beaker where I put it to where it's low. There was nothing in the cell itself, so that iodine moves into the cell, interacts with the starch that is in the cell, causing that